All right. Thank you, Bioanalytics. Thank you, Bro, for hosting us. So my name is Weng, and this is Joe. He will be doing most of the presentation today. But before we get there, just a disclosure for the presentation. So I think before we start, just a quick introduction. So we are part of the Sanofi mRNA Center of Excellence. What I think you can fully appreciate that, a lot of the work that we do is so fast paced. Sometimes we don't even have a story to tell. So I think I really want to appreciate, our department and our legal team to let us tell you whole story of what we're doing and how Spectronaut helps us to basically build this story and basically make decisions go and no go for our phase 1, phase 2, phase 3 assets. So this is basically, I think this slide is a couple of years old. These are all the technology platforms here at Sanofi. I think to date we have more than 20 different platforms, from large molecules to small molecules and to oligos. So the mRNA Center of Excellence basically work in mRNA vaccines. So it's probably not the most popular topic in this country right now. So there are actually two different platforms in there. There's the mRNA designs, and then there is the LNP, which is the lipid nanoparticle formulations. So this is what the mRNA COE basically works on, these two modalities combined into one as mRNA vaccines. So here in Waltham site, so our facilities is based in Waltham, and the whole organizations of the COE is about 550 full-time equivalent staff employees. We go end to end from discovery to analytical development, all the way to translational science, manufacturing, and to patients. So our team is actually embedded in the translational science. We provide all the high-end mass spec characterizations from mRNA designs to how dose formulation looks, provide high-resolution mass spec analysis on your drug substrate, drug products, and then we have access to clinical samples just to understand what's the immune response of those assets in patients. So, so we are the bioanalytical team. So what that means that, on top of doing proteomics, so my team oversees all the in vivo biodistribution studies in mice, in rats, in NHPs. We do lipid characterization stabilities, thermostabilities. So I have team members for that. And then, I'm classically trained as a proteomics person, a mass spectrometrist. So they brought me in. They did not have any discovery pipeline or workflows to look at global proteome changes. So since I've joined the COE, so we now have basically all of these capabilities. Of course, we do a lot of on a daily basis, do a lot of peptide mappings. And we do this using Spectronaut as well. So that just tells you how versatile the whole software is. So the three stories we kind of want to, have the green light to basically share with you is basically looking at and understand how each of our formulations attracts different biological fluids onto the LNPs. And then based on that, how it gets distributed to the whole body. The second story that Joe is actually showing today is our unconventional use of Spectronaut, basically to understand non-canonical translations from all the different mRNA designs. The third story is we don't have the data yet. All our samples are still in Schlieren. Is basically because we are in vaccines, so they want to understand if any of our mRNA designs get presented on the APCs, mostly HLA-2, and that will inform whether a design is good or not good. So in the interest of time, so what I kind of want to plug is that the first story is a full story, so basically showing you over 60, 70 different formulations. We basically mapped out how our formulations attract different binding of proteins. And this will be shared next month in collaboration with Bioanalytics. We will be hosting that. And I think Bioanalytics is going to be also giving a talk on the launch of SpectroNet 21, which is no, those people out there. They will be giving a talk basically launching the SpectroNet 21 pre-ASMS. So with that, before I hand it over to Joe, I just want to point out that this talk was actually, I've presented this many different iterations. This was part of a bigger talk at Sanofi. It was part of the AI and ML machine learning talk that we basically showcase all the different machine learning designs of mRNAs. And today we are happy to share a small part of that talk with you. Here you go, Joe.
So hello, everyone. As Weng just mentioned, my name is Joe McLaughlin, and I'm a research associate at Sanofi's mRNA COE in Waltham. And today I'm going to be presenting on some of the work our team has worked on regarding ribosomal frameshift events. So as we all know, our DNA transcribes into our RNA and then translates into proteins. And coming from the mRNA vaccine industry, our mRNA is going to be that main modality. But however, we cannot just inject mRNA into the patient. mRNA is very unstable in the body, so we have to utilize an encapsulation method, so we use lipid nanoparticles, or LNPs. And basically, for the last 15 years or so, a lot of research has been poured into this field in order to try and create RNA strands that are more stable. So these two papers shown on the screen right here played a monumental role in getting the RNA therapeutics to where they are today. So this first paper on the left comes from the University of Pennsylvania, from Carrico's lab. And basically what Carrico showed is that if you modify the nucleobases within an RNA strand, specifically the uridines into a pseudouridine, that we actually are able to observe enhanced protein translation and overall stability in the body. And Sonnenberg's lab, in a collaboration with Moderna, was able to take this a step further and showed us that this is actually due to the fact that there's an increase in ribosomal density on these RNA-modified strands. And one thing that is important to note is that these modifications should never take place at the 5' cap. This is where our ribosome is going to dock onto our RNA strand and begin that protein translation. However, if we have those pseudouridines in this cap, pseudouridine is very bulky and it actually can hinder that process from taking place. So all the RNA therapeutics that are on the market in today's day have a proprietary 5' cap that is engineered to kind of promote that ribosomal docking and overall downstream translation of our proteins in the most efficient manner. So moving forward, in this paper from Bioanalytics, it also showed that these modified mRNA strands also work to decrease our innate immunity in the body to the free RNA. So free RNA normally can be recognized by TLR receptors in our body, which trigger a downstream immune response and basically flag that RNA to tell the body to get rid of that. So more specifically, the uridines within the normal RNA will be recognized by the TLR7 receptors, which initiates that immune response. However, when we modify those uridines into the much more bulky pseudouridine, that recognition between the receptor and the base is now hindered as well. So this leads to an enhanced overall stability of our RNAs in the body and also can contribute to an increased lifetime of the RNAs in the body as well. So moving forward into 2021, Sonnenberg also kind of built on his previous research. He showed us that the reason for the increased ribosomal density when we have these pseudouridine modifications is actually due to the fact that ribosomes will stall mid-translation at these sites. However, he also showed that when this happens, the endoplasmic reticulum membrane within our cells will come in and kind of support that ribosome and help push it through those translational spots so that we can continue translation. And all of this built up to pretty much the most significant paper to come out in the last couple of years in our field. And this paper was published in a joint collaboration from AstraZeneca and Oxford. And basically what this paper reported, it was the first paper to report that the Pfizer COVID vaccine, which we all know, actually shows evidence of a +1 ribosomal frameshift event. And you might be wondering to yourself, what is a ribosomal frameshift event exactly? Well, basically how our ribosomes work, they translate their RNAs from the 5' end to the 3' end, reading all of the nucleotides in the strand in groups of 3 called codons. And basically it has that main reading frame on the antigen where it goes down and reads from 5' to 3'. However, if we were to shift that reading frame +1 unit or -1 unit, we now are going to be translating completely different codons downstream. which can lead to different amino acids being built onto the protein and ultimately a completely different protein being synthesized than what we originally wanted. And the way that we kind of make an analogy with this is that we kind of think of it like a zipper. Like usually your zipper just goes up and down pretty nicely, but sometimes it can get kind of caught, so you kind of have to yank on it. And when that happens, sometimes the zipper can become misaligned and we have a completely different product. And this is kind of analogous to what happens when a frame shift occurs. And obviously coming from the mRNA therapeutics area, we never want to be translating a product that is not intended in the body. So this kind of brings us to some of our work at Sanofi. To this date, pretty much all of the in order to be able to screen for frameshift products, we have to rely on cell-free translation and libraries. And pretty much, if you've ever worked with libraries, it's a very laborious process. You have to manually annotate all these different spectra. And the fact that it's also cell-free, means that we are completely eliminating the cellular proteome aspect. And coming from the mRNA-CoE, we are generating hundreds of different mRNA strains every week. So this overall process is just pretty much infeasible for us. So we wanted to create more of a novel assay that can screen both our frameshift products and be able to monitor our cellular proteome at the same time. So with the power of Spectronaut, we were able to do just that. We've created this library-free DIA workflow. So the beginning of this workflow works the same as normal. We just do our usual sample prep, which I'm sure you all are familiar with. But the beauty of this workflow actually comes from an in-house tool that we have created that can actually go through an antigen and go through every single nucleotide in that antigen and simulate a frameshift event occurring. And then what we also are able to do is figure out what that protein would be when that specific frameshift occurs and add it into a theoretical database. And as you can imagine, we repeat this for every single possible nucleotide down the line. So there's going to be hundreds, if not thousands, of permutations for each antigen. And we compile all of those into one database, and then we add that database into our known human proteome database. So now we have one comprehensive database with all the possible mutations of our antigens, as well as the entire human proteome. And for this study, we tested this on 5 different antigens: our RSV, our HMPV, our influenza A strain, our influenza B strain, and a machine learning optimized sequence. For the sake of this talk, I'm only going to be discussing our RSV, our HMPV, and our influenza A. So basically, we're looking to figure out the answers to 4 different questions. We're trying to figure out if we can see expression of our antigens. We want to see at what level our antigens are being expressed as well. Obviously, that's the main point of our mRNA therapeutic, is to be able to showcase that antigen is being expressed at a high level. We also want to see its protein coverage, and we also, for this experiment, wanted to see if we can observe any frameshift events. So if we take a look at our rank protein abundance graph on the left-hand side, we see that we have positive signals for our flu A strain, our RSV, and our HMPV. We also do detect a potential frameshift product of our influenza A strain, which I will get to in a little bit. This was all done on Explorers 480 on a 90-minute gradient. We have about 115,000 precursors, 96,000 peptides, and about 7,500 proteins. We recently just repeated this experiment on an Astral, and we're able to increase that protein ID number about 20%. And now if we point your attention over to the table, at the top of this table we have our GAPDH, which is our housekeeping protein. This allows us to normalize data across different experiments. And you might be wondering, under the protein coverage, why all the peptides in the protein coverage for the GAPDH are coming green, whereas our antigens are all coming red. And if you remember how we designed the database that we're using, we have thousands of different antigens with all the different permutations from those frameshift events. So we're going to have a lot of different carryover of the peptides across all those thousands of different antigens. So in reality, they are unique peptides, but just the nature of our database they're going to come back as ambiguous. And the key takeaway from this slide, though, is that we do not detect any frameshift products in our HMPV or our RSV. Recently, we also ran an experiment utilizing proteasome inhibitor on these strains, because we were wondering that maybe they are having frameshift events, but our proteasome is just coming in and eliminating them right away. So we wanted to inhibit that proteasome and repeat the experiment. And I'm happy to report that we still do not see any frameshift events even after the proteasome is inhibited. So moving forward, as I mentioned, we do detect that frameshift event in that influenza A strain. So if you, at the top of this table is our main reading frame of our influenza A, and we have on the right-hand side the peptide signature of that influenza A. And we detect two potential frameshifts from that influenza A. And if you go to the signature right here and just go along this, along this footprint, you can see it's the same sequence all the way until about right here, that residue 461, 462, where we detect a +1 frameshift in our FS1 product, which ultimately changes the protein downstream. And we can identify this peptide, which is unique to this strain, downstream. And the same thing is true with our FS2 product. We have the same footprint all the way up until this 529 through 584 dark zone. And we can see this peptide at the end, which is not present in the main reading frame as well. And you might be, and I'd like to now point your attention to our log2 abundance. So you might be noticing that these numbers are pretty low. For our workflow, we need to be able to provide our CMC teams downstream with a relative quant. So this is just a relative quant for them so they can have a kind of a benchmark to go in and do a more targeted workflow, an absolute quant. And they also want to be able to have a percentage of the main antigen. So we report that the FS1 shows up 0.31% of our main antigen, and our FS2 product 0.38% of our main antigen. We also are going to provide the CMC team with our MSMS footprint. And for this, we really want to show that all of our peaks are coming out at the same time and that our Y and B ions line up. And it's because it's such a low signal, we want to make sure that this isn't coming from noise or an artifact in the software or noise from the instrument. And because this is pretty significant data to be sharing with our project team, we also want to validate this with an orthogonal method. So for that, we used RiboSeq profiling. And the way, what we want to see from our RiboSeq profiling is that our main reading frame, which is that blue color here, is the vast majority of the reads we are getting. And I'm happy to, or Flu-A, it definitely shows that blue color for the vast majority of the time. However, if you zoom in around the 1500 mark, about 1480, we do detect one out-of-frame read in that red color that's higher than that blue main reading frame. And this zone actually corresponds directly to our previously reported potential frameshift event that we detected with MS, and that frameshift 1 product. So that's around nucleotide 1480, and our amino acid residue is around 460. And this starts at the way that we set up the RiboSeq profile, that is where that would show up in the ribosome, or in the nucleotide positioning. And then we also showcase, for that FS2 product, we showcase an out-of-frame read that's following the stop codon. So this black line is our stop codon, but yet we're still detecting reads from after the stop codon, which is more evidence of a ribosomal frameshift event happening. And again, the key takeaway from this slide, though, is that we repeated this with our HMPV and our RSV strains as well, and we still did not see any frameshift events for those strains either. And now we are currently, as Wayne kind of mentioned, working with Bioagnosis to do some immunopeptidomics to make sure that that potential frameshift products aren't being exposed on the outsides of the cells, and HLA-2 antigen presenting cells, I should say, because they could be, in theory, being made within the cells and then immediately being taken out by the proteasome and then potentially being expressed on the surface. So we just want to validate and make sure that that's not what is occurring. So in summary, we have developed an in-house tool and a library-free workflow to generate de novo database for frameshift products and be able to identify and quantify endogenous frameshift products. We detected no frameshift peptides for our RSV and HMPV strains. We do detect trace evidence of frameshift events in our Flu-A, and we used RiboSeq profiling to show minor out-of-frame reads, which can indicate potential low-level frameshift events. And with that, I'd like to say thank you all for your attention and for Bioagnosis for hosting. And I'd like to thank everyone at Sanofi for making this possible. Thank you.
Thanks for the talk. Just to clarify, when you talk about the de novo library, do you mean like you include all 6 possibilities of the frameshift to construct your protein reference database?
More than that. So you have for each antigen, you have your three open reading frame, the main ones. And then we basically go on nucleotide, every single nucleotide, when you shift one or you slide back one, what is the resulting proteins?
So for that for the single vaccine, how big of the search universe?
A single vaccine or single antigen,
Yeah.
it's about just about over 1,200 possible permutations. But you have to understand that some of them have a premature stop codon. Some of them, they don't really qualify as a peptide even. So we have to filter those out. And I think once you do all these manual curations and look at what are the possible peptides, and then they kind of come to around about 1,100.
And do you use the trypsin for the cleavage?
Trypsin, chymotrypsin, we've done it all.
Okay, thanks.
You're welcome.
Great talk. First of all, kudos for pushing that through legal. Impressive. My question is, when you do have that trace amount in like below 1%, is that something that's considered that eventually that could be allowed, or
So
are there?
yeah, so I mean, we presented this talk in the analytical forum. 1%, it's a red flag. Even 1% is a red flag. So we hope, I mean, anything less than 1, I think we're on the right track. But if we're hitting around 1, it's a big problem.
So the 1% would be kind of the cutoff where, like, if we are hitting that we need to kind of figure out how to
we'll change the design.
Yep. And if we are, let's say, 0.01%, we say, okay, we can continue.
Yeah, so that's one thing, right? I mean, like, we are all with Spectronaut, we are hovering like way below like the LLOQ anyway. So I don't think anyone of us here can basically make a call on the absolute quant of a peptide. So this is where we do the hands off to the CMC team, like the process development analytical team, to actually go in and do the absolute quant. So further de-risking the asset.
Thank you so much.
Back there.
Quick question here. So, can you comment a bit that for this type of analysis, whether RiboSeq data is preferred? Do you really need the mass spec data? And also, for the type of task, it seems the MS1 intensity could be also looked at, right? Because that is really peptide specific and you don't have the window size consideration.
Okay. Yes, RiboSeq is preferred, but in terms of throughput, we're looking at hundreds of mRNA designs a week. So we can't do RiboSeq for all of them. Anything that we have positive hits, yes. Secondary validations, RiboSeq. I don't know if that makes sense. Yeah. The second question's MS1. This is a touchy subject, right? Because, I think analytical team always comes back to us about quant. I think with this type of workflow, discovery workflow, and all your signals are below, I don't know, if you use Spectronaut quite a bit, anything below 15, like you kind of like question, like log to abundance, like questions like if they're real quant, relative quant, whatever. So I think the red flag, I mean, not the red flag, the one considerations when we pass it down to the analytical team, like really, they can do a much better job at quanting this than us on the discovery workflow.
My question is regarding whether these novel epitopes that might be escaping based on the Frameshift, whether you can predict if some of them are undergoing like nonsense mediated decay, or that's the idea of this approach. I don't know.
So I think the biggest thing that we are doing right now is basically trying. So AstraZeneca's paper, that was fantastic. I mean, that was basically launched the whole investigations within our department. So they proposed a certain motif on the codon designs, like basically uridine, and then something else will promote Frameshift. So the machine learning team's actually taken that into considerations in designing stuff. So right now we're in the process of basically proving or disproving that is the actual what we see. We're work in progress.
Thank you very much.
Thank you.